Okay, now after iron and boron, what we are going to take next is manganese, copper and zinc. Three of these are going to be considered. Now, you know these ions or these minerals are very important in terms that they are activator of certain enzymes. If you can't remember anything, for instance, what you have to remember is manganese, copper and zinc, these three are very important cofactors in enzyme activation, and you can very well realize if something is important for enzyme activation, how crucial that particular element would be, because enzymes are the ultimate catalysts which are responsible for which are responsible for enabling the carrying out of various metabolic activities. So let's take into account manganese first of all. How it is absorbed from the soil? It is absorbed in the form of bivalent ions from the soil. It is present in form of, not just in the form of bivalent ions, it is present in the form of tetravalent ions as well, but it is not possible for the plant to take up these manganese ions in those forms, so it prefers taking in bivalent ion form. Now what it does after it has been taken up by the plant, it activates several enzymes which are important for nitrogen metabolism, and we know that nitrogen was one crucial macro element. So this particular element is going to play an important role for activation of enzymes like nitrogenase. So if nitrogenase, we discussed that if nitrogenase is not present, nitrogen uptake would not be there. So in case the manganese deficiency, there would be deficiency of nitrogen as well, which would be observed by the plant. So because manganese is important for nitrogen uptake, the deficiency symptoms would be somewhat similar to those of nitrogen, but they would not be as much aggravated as in the case of nitrogen, but they would be there. So first symptom that we come across is chlorosis would be there. Of course, the green part of the plant, that is the leaves, they would be turning yellow, followed by death of the particular plant part, necrosis, and this chlorosis begins in interveinal areas, due to which the leaf appears that greenish reticulate fashion is observed in the leaves, that you would find the veins and the midrib, they would be green and rest of the leaves would be yellowish. Now another important factor, that is the most important, the one which you should remember, one which is of importance of manganese, is that it plays an important role in the photooxidation of water. Now what is photooxidation of water? Another term that we use for it is photolysis. Lysis means breakdown of something and photo means in the presence of light. Now which light plant takes? It takes the sunlight. So in the presence of sunlight, what happens is the water molecule breaks down because the energy from the photons is taken up by a particular component of the leaves, that is a photosystem. It gets electron deficient, and this water molecule, it breaks down to give that electron deficiency and overcome by giving the electrons to that electron deficient photosystem, and in the process it is broken down, and this is the source, this process is the source of all the oxygen we take up. Okay, this process, that is the photooxidation of water or the photolysis of water, is the process that gives rise to the molecular oxygen and enables your and mine survival on this earth, because we need oxygen in great quantity. For every time we need oxygen, we cannot survive with oxygen. So you can realize the importance of this particular process, and this does not take place if manganese ions are not present. Now the next point of importance is that it is also important for synthesis of chlorophyll and oxidation of oxins. Okay, indole 3-acetic acid IAA cannot be oxidized without manganese ions coming into the scenario. So we get to know that it is important for synthesis of chlorophyll, it is important for photolysis of water, it is important for the activation of enzymes which are crucial in the nitrogen metabolism. So you can make it out that this micro element is of great importance to the plant, and whenever it would be deficient, the plant would show chlorosis in the beginning and followed by necrosis. Okay, this is what manganese deficiency is going to cause to the plant. Next we come to copper. Now please remember one thing, whenever we are discussing copper, if the soil is rich in copper, that means it exceeds a given value, it is toxic for the plant. Okay, so what is important for the plant is that less than 1% of copper should be dissolved in the soil solution, and then plant would be able to take up that copper in the form of that dissolved solution itself, and it should not exceed a given value because it is toxic. First point of remembering. Second is that you might have heard of this term in your earlier classes. If you haven't, I am making you aware of this thing, that plastocyanin is an important electron acceptor. Okay. Now, what role these electron acceptors play is that I told you that sunlight is taken up by the leaves, that energy is taken up by leaves, some particular photosystem loses its electron. Where that electron goes, that would come to the electron acceptors, out of which plastocyanin is an important electron acceptor when the excitation takes place in the presence of sunlight. So, this plastocyanin is important, and this plastocyanin is formed by copper. It is an important component of plastocyanin. So, you can realize the importance of copper, that it would be very important for carrying out the process of photosynthesis. Then it is activator of certain enzymes, and specifically oxidases. So, oxidases are those classes of enzymes which enable the oxidation. Oxidation is the either gain of oxygen, or in terms of proton, you know, proton plays an important role. Both this and electron. These play an important role in electron transport chain, and so do enzymes which help in the transfer of either the electron or the proton from one entity to the other. So, it is activator of oxidase enzymes. It is a crucial micro element. I am sorry, it is not macro element. It would be required in very less quantities, but yes, it would be required. A plant can't do without it, because you see it as part of plastocyanin. Most of the enzymes would be activated in the presence of copper itself. And what if this copper is not present? Distortion and chlorosis, which would be followed by necrosis, and even complete plant death can take place, because there would be no growth that would be observed. So, we have distortion and chlorosis. One point of remembering over here is that it is shown in young leaves. I have told you earlier as well that you need to remember which thing shows what symptom in which part of the plant. So, this copper deficiency, chlorosis due to copper deficiency would be observed in young leaves, which would be followed by necrosis, and ultimately complete plant death can also take place. Now, coming to zinc. Zinc is very important for the plant because it is synthesis of tryptophan. It is important for synthesis of tryptophan. Okay. Tryptophan cannot be synthesized in the absence of zinc. What is tryptophan? Tryptophan is precursor of auxin. Now, what is auxin? Auxin is the plant growth regulator, which is very much required for the growth of the shoot apex. You must be knowing that shoot apex develops if auxin is present. So, this tryptophan cannot be synthesized in absence of zinc. If tryptophan is not synthesized, auxin will not be synthesized. If auxin is not synthesized, how can you expect a plant to grow normally? So, basically zinc is very important. Coming to the most basic thing, that how this zinc is absorbed from the soil, from the withering minerals, you would find divalent zinc cations, and the plant would take up those divalent zinc cations as it is from the soil. Now, it is activator of enzymes. Which enzymes? You might have heard about carboxylases. Again, an important class of enzymes which play a crucial role in the processes like Krebs cycle. They are important, mostly important in the case of respiratory processes. And of course, there is an enzyme known as Rubisco. You might have heard about it. That is carboxylase only. So, wherever the carboxylation process would be required, the enzyme carboxylase would come into scene, and if enzyme is coming, the activator zinc would be required. Okay, what happens if zinc is not present in the soil in appropriate quantity which a plant requires? First of all, auxin would not be synthesized. How can you expect the nodes to be of the appropriate size that a plant should have? So, we observe that internodes are shortened. If the internodes are shortened, by default the plants would have stunted growth. The internode elongation does not take place, so the plant would be stunted, as other processes would be hindered. So, there would be reduction in leaf size itself. okay, leaf size would not be there, because carboxylase is an important enzyme required for the metabolic process of photosynthesis. So if the photosynthesis is not taking place properly, how can you expect the growth to take place? And the reduction in leaf size points towards the deficiency of zinc, and because all this is taking place, there would be intervenal chlorosis followed by necrosis, and this would be observed in older leaves. This won't be observed in younger leaves, this would be observed in older leaves. So this is the role of all these three micronutrients. Next we are going to take molybdenum, chlorine and nickel, but for once you need to remember that all the three are important microelements which play an important role again in the activator of various enzymes, which are yet again important for various metabolic processes. Thank you.